We do have a couple of audience questions here that I want to throw to the panel. We've got one that's, what are the areas in either up or downstream process that represents the greatest opportunity to increase manufacturing efficiencies? I don't know who would really like to any of you want to tackle this one? share any wisdom on how to make this fast process even faster? Jeff's laughing. Not to put you on the spot, Jeff.
I'm laughing because I think it gets into some things that maybe we'll talk about later, and that is, the kind of the QBD aspects, and really this is still a relatively new technology. Yeah. The IVT reaction is pretty straightforward, but there's a lot of optimization that can be done to figure out, what are your best yields, and that can only be done, through DOE, right? Just you got to go through the grind and figure it out. Again, there's a lot of good work that's been done out there, and we can leverage that. But in the end, for your construct, whether it's linear, and I am speaking linear, right? So that process is pretty straightforward. You have to work through it and figure out what the optimal conditions are for your construct.
Yeah. Jim, do you have anything to add from that circular RNA side of things?
Yeah, no, I agree. I think, one of the great question in terms of efficiency, right? And I think that's where IVT, again, fairly straightforward. It can all be addressed, in a synthetic type of process, parameter setting, DOEs. But I think what we need to, obviously, from a downstream perspective, as we learn, as we understand what sensitivities when we get into therapeutics, where we need to push our purification, what the final quality profile looks like, I think that's where we're going to have to obviously, make headway there. But I think really in terms of efficiency, it's not always productivity. I think that's one thing we need to remember is that people quote a whole single digit, low teens productivity out of an IVT. Is that clean? If you drop to 6 grams per liter rather than putting it, being excited about 8 or 10 or 11, right? But you're generating additional double-stranded, additional truncated, additional any sort of other impurity. The efficiency economically, you need to find the right balance. And to Jeff's point, that's how that's where you need to understand your DOEs. There's multiple different outputs that you need to pay attention to from a manufacturing and scalability perspective, because you end up moving the bottleneck somewhere else. It's the same monoclonal issue we had 20 years ago, right? You can make 20 grams per liter, but you can't really get a column big enough to purify it. So it's a balance. And the whole industry is looking at this, and I think again it's come up already a few times, is getting away from a low dose vaccine, 1, maybe 2 or 3, boosts, and replacement therapies, whether it be monoclonals or enzyme replacement. We're talking about potentially lifelong treatment. So, and at a higher dose, depending on the target tissue or cells and everything else. So I think it really is understanding what your efficiencies need to be at the stage you're at, and then really kind of think forward to how much can we optimize this as an overall process. So it's one place that you can't, upstream and downstream are tied very tight together, as is the whole drug product or LNP process as well. So definitely an area we need to focus on. But I think patient safety is probably the key thing. So don't push your efficiencies beyond what
too far.
Exactly.